Welcome to the 30th Emeriti lecture. Boy, I can barely see people out there. I'm Todd Whipke. I'm president of the Emeriti Association, which is the organization that sponsors this lecture series. This lecture series was started by Joe Burnett some time ago, and its purpose is to highlight research performed by retired faculty after they have retired. Many emeriti remain quite active. In the past 3 years, 62% of emeriti are mentors to students. 36 are still teaching. 60% have published one or more research articles in a journal. 15% have published a book, one or more books. Emeriti brought to campus $17 million over the past 3 years and donated to university $1.8 million individually. So I want to thank Chancellor Blumenthal for sponsoring funding this series, and Alicia Burse for helping to organize this, and other staff that are making this possible. If you would like to see, this is video recorded, so turn your phones off. And if you'd like to see previous talks that are recorded, just search for UCSC Emeriti Lectures on Google or on YouTube, and you will find the whole series. A little advertisement, please click on subscribe when you're at YouTube. We would like to increase the number of our subscribers, and you'll be notified of new material. So our Emeriti lecturer tonight is Distinguished Research Professor Phil Cruz of Chemistry and Biochemistry, a colleague of mine, and he's going to speak on inspirational molecules from Oceania. Two of Phil's former students are going to introduce him, and I'll introduce the students in the order that they're going to appear. Tyler Johnson, who got his PhD with Phil in 2008. He was an undergrad here as well. He went on to do a postdoc at Berkeley and is now assistant professor at Dominican University of California in San Rafael. Sarah Robinson got her PhD 1 year later, 2009. She did a postdoc at Northwestern and is now a scientist at Genentech, working in the area of small molecule structural elucidation. So Sarah and Tyler, please introduce Phil.
Thank you, Todd. So Sarah's going to do the technical work for me while I attempt to get my act together here. Okay, so thank you all for coming. Thank you, Todd, for the introduction. It's a remarkable career Phil has had, so he needs two graduate students to actually introduce him. I'll be introducing him from the 1950s to 2000, and then Sarah will segue from 2000 to 2019. So you'll all have probably seen this somewhat similar flyer here that we've circulated for the last several months talking about Phil. Bottom line is he's going to give you a talk on this area right here, and at this point he has some 242 odd scientific publications on biologically active marine natural products and biomedical research. And so, being with Phil all these years, I can just give you the very stale rundown of his resume or his CV, but I wanted to give you the Hollywood true story and highlight some factoids that are unable to be accessed online. And so when you see these asterisks in red, you'll understand what I'm referring to. So basically, we have to talk about, unfortunately this got deleted in the Mac PC conversion world. This should say pre-CV right up here, okay? So pre-CV, Professor Cruz actually got his high school degree from, Cubberley High School back in the day in 1959. Here's Phil as a sophomore, and another colleague at that time was Professor Will Fennicle, who was also a marine natural products expert. And I want to highlight this because Cubberley High School was started in 1956 and went away in 1979. Well, times change. Got to change with the times. So Phil got his roots prior to coming here, right over here in the Bay Area. And so here are some selected examples of early milestones in his CV. He got his bachelor's degree at UCLA in 1966, his PhD only 3 years later at UC Santa Barbara, back in the Outback with Dominic Bertelli. Did a postdoc for a year with Kurt Mieslow, and then he came on board in 1970 to be an assistant professor at UC Santa Cruz. One of the E-Hollywood true stories that you didn't hear about is he was also offered a job at Cal State University, Sacramento. How this discussion would have been so different if he had his actual long-term career at CSU Sacramento. So keep that in mind. And going forward, some of the asterisks are actually missing here, but the stay tuned part refers to his access at University of Arizona, and then we have a discussion later that you'll hear about with regards to his textbook production. So, wow, these slides have been changed on me, so I want to look to the people who've adjusted these without me being aware of what's going on here, because I'm kind of in the situation not knowing what the hell is going on here. But basically, at the end of the day, Phil was an assistant professor at University of Hawaii in 1974. He got tenure in about 1977. There you can see he's an associate professor. He was an assistant professor at University of Arizona for 1 year, for 1 semester, or 1 quarter rather. And I don't actually know what's going to come next in the slides because I haven't had a chance to see these edited versions, so just be patient with me. But some of the major milestones would probably be in the early years from 1970 to 2000, the addition of this incredible textbook. So as a show of hands, how many people here today are assistant, associate, or full professors in academia at a university or college? Please let me raise your hand. So by eyeball integration, that's about 25%. How many of you have gone on to publish a textbook? So it looks like 1 or 2. So why is that relevant? This is a significant milestone. And I ask these questions because I asked Phil when I was a graduate student, how the heck did you get a paper published when you were going on expeditions you're going to hear about and publishing scientific papers? Well, I'll never forget, as a graduate student, is what he told me. He said, Tyler Johnson, Jaime Rodriguez, Marcel Jaspis, we would get up every Sunday morning. We'd meet at 7 a.m. and we'd discuss at 7 a.m. every week for a year how we're going to make contributions to this. And that's how this book came out. So the first edition came out in 1988. The second edition came out in 2008. So incredible milestone there I want to highlight. Not sure what's coming next. So basically, now I'm going to segue to my colleague here, Sarah Robinson.
All right. Thank you, Tyler. Okay. So if you look at Phil's CV, it's clear that he really had a knack for getting funding. He looked to a number of different places and was really valued for the critical feedback that he gave on funding committees, and was incredibly successful at keeping a prolific research group and funding undergrads and research staff throughout his entire career. I think this is benchmarked by the number of manuscripts that he was able to publish, the books he was able to put into publication, and over 70 grants and awards, 31 boards or professional committee appointments, 8 patents. I mean, it was just, if you look at his CV, it spans about 11 pages, and it's definitely worth a read. So I think some of the more important stuff to highlight, though, is just how many people he has mentored throughout his career. And he calls them colleagues or coworkers and things that you learn. It's a mutually beneficial relationship. But I can attest that the mentorship we got there is really unmatched. And 260 people have been fortunate enough to be mentored by Phil, myself included. And that's not including the Access students that rotated through his lab, the MBRS and MARC students who he created adventures in bioorganic chemistry for every summer, and the hundreds and thousands of organic chemistry graduate students and wine chemistry students that he taught through UCSC. And I want to stop right here and point out all the work he did in funding these programs to give scientific research access to underrepresented minorities. And the amount of time and the successful funding should be celebrated right now again, because he just got a 17 and secured 5 more years of funding for the Access program. And I want to say that, that at the time, we had tons of underrepresented minorities coming through our lab every summer, but that isn't where it ended. It was the way Phil showed up at conferences, and it was the hiring and people he brought into his group that really, across the board, this is who Phil is, what he believes in, and what he's committed his research career into promoting, advocating for, and providing opportunities for. And I had no idea how important that was to producing good science until I left the lab and recently worked on a project team where, again, at the table, it was filled with a diverse group of people, whether it's ethnicity, religion, anything. What I find in that environment is that the only thing that matters is the science. There is no majority, there's no barrier to speak up. It's truly just truth in the science. And Phil, more than anyone, I can't thank you enough how that's a core value of mine as a scientist. And I think amongst everyone who's worked for you, that's something that we've really come to appreciate over time. This is Phil's day, and so I asked him a few questions, and I didn't give him very long to answer or very much space, but the first was really, what was the best part of your professor career? And obviously, it's getting assistant professor at UCSC and overcoming the big barrier to tenure. I think the hardest part was getting tenure, but as you can see, with all the funding he was able to secure, that was easily offset. And lastly, what was the most meaningful impact of your career? And of course, he turns the spotlight to those around him and says it's the more than 260 coworkers he was able to spend time with throughout his career. And lastly, I asked a few of the former graduate students some of these same questions. What do you think was Phil's favorite part? Telling people, exactly where they should go with their careers. Bringing people to the table with stories, making pizza for people, pouring wine for people, really just engaging in conversation. And I think that defines Phil. Also his lifelong commitment to helping colleagues, and really the academic freedom that he's been able to establish at UCSC to drive collaborations and impactful research. What did he dislike the most? Aaron kind of nailed it, box wine, downtime. I think that made a couple appearances, along with grading. And most significant impact Phil has had on your career, all the opportunities he's provided for us, definitely all the travel, the different cultures and the diving. I don't know, there's a few personal notes in here, but also the importance of asking a question, and understanding the impact that can have on provoking thought. And lastly, the most significant impact Phil has had on the field, and a lot of that is in the chemistry. So jasperkenolides and bangamides, I think Phil's going to talk more about that today, and the bioactivity that they possess. Again, great chemistry and commitment to training students. And Phil, recently, we're talking about contractors and different people to hire, and Phil really does think about second chances and opportunities for those that may not always have them. I think that's signature of the, impact he can have on those around him. And also just the impact of all of his collaborations he's been able to establish over the years. I think, with that, Phil, we're all incredibly thankful to have had you as our mentor and are excited to hear some more about the science today. So please come on up.
organized here. Todd, I agree. This is like being in an interrogation. It's hard to see what the heck is going on out there. So, all right, here's a problem. About a year ago, Todd Whipke, who is our president of the Emeriti Association, sent me this email to invite me to give a talk. And so the email came, and I just let it sit. I figured just let sleeping dogs lie. Then my colleague Claude Bernasconi sent me a reminder email. Did you get Todd's email? And, get your act together. So finally, I responded because I knew April 9th of 2019 would never come, and I'd never have to worry about doing this. Well, then as I began to think about the instructions that Todd had, finally looked at the email, and just recently I looked at what the marching instructions were from the late Professor Joe Burnett when he helped to set this up. And the idea, in fact, let me just mention about Burnett. May 3rd is the 19th annual Burnett Lectureship. It'll be over in PSB building, and I invite all you folks to come. It starts at 7, and it'll be a glorious celebration that will continue sort of an Oceania theme because my colleague Bill Gerwick will be giving the keynote lecture. At any rate, all right, so back to this business. I couldn't come to grips with the restriction of just talking about what I had done since I retired, but I don't like this retired word, and I don't understand what it means. And from the standpoint of the comments that Tyler made a few minutes ago, it seemed to me my job was to get tenure at UCSC, and in the old days, in the 70s, this was the Wild West. And so eventually that happened. I crossed that bridge, and then the sense of a job went away. So I'd gotten tenure. I didn't have to have a job anymore. And if you don't have a job, you can never retire, and you never become an Emeriti person. So that's more or less my situation. So I can't apologize. I'm just going to tell you a bunch of things that we've done over the years that have kind of started in the older days, but continue today. So that's the theme of what I'm going to do here. All right, let's see. The pointer works. Organic chemistry is biomolecules. I should have come up with a better title because it scared away most of my humanities colleagues. And but nonetheless, we'll carry on. So these are some of our favorite molecules. The bengamides that Tyler just talked about a minute ago, and Madeline Adamczewski is here. This was Madeline's thesis work decades ago with Emilio Quinoa. Madeline, is Emilio here somewhere, and he'll appear in a minute? But maybe not. At any rate, here's another molecule, Cimbrin. So molecular structures, this is the language of organic chemistry. And so I'm going to have to parade some of these molecules out on the floor. And those of you that are not so inclined to kind of engage yourself in looking at these things, just think of them as patterns. All right, now there's another cautionary thing. Sometimes it's hard to look at these patterns. The ushers are prepared to travel the aisles and give everybody aspirin pills in case you want to have some help from chemistry to get over that part in terms of looking at all these structures. But anyway, all right, let's journey forward. And so just to give you a snapshot of what's been going on in the lab for decades. All right, so our goal, our overview, is to really look at novel small molecules from the standpoint of having the ocean as our playground for discovery. And at the same time, what we're trying to do is to define new principles of how these molecules are put together. In other words, these molecules have handedness, there's enzymes, there are things that are involved, and a way to get entree into that subject is to have a sense of using the molecules to really define that process. Expeditions. There's no such thing as a theoretical natural products chemist in my mind. And so to begin the journey, you've got to go out into nature. And so it's our continuing goal to go to areas of high invertebrate biodiversity. We've tried to emphasize also biogeographical studies on sponges that I define as chemically rich, and we'll see some examples of this as I go forward. Using the sponge microbiome is a twist that we've sort of engineered and we've approached in our program in the latter years. So that's another platform for our work. We want to transform the leads we get into something of therapeutic usefulness or molecular probes, and I'll show you some examples of that. We want to employ new modern methods, next-gen methods to essentially, especially with the microorganism culturing work, to expand the overall scope of the project. And to look from a standpoint of once we have an understanding of the various molecular structures, to be able to put these on the table to look at questions in other areas. So the work really starts with sponges, it translates back into a molecular structure, and along the way, we've been able to use the screening center that my colleague Scott Loki set up many years ago. And these are a series of phenotypic screens that we can get from the hand of Walter Bray, who has been the person involved in that laboratory for the last decade or more. Now, so we had a report here from two former group members. Tyler was first, and Sarah was next. This is a photograph that was taken in 2007, and there's some explanation that needs to go on here. Both Tyler and Sarah were graduate students at that period. Tyler was, we were pushing Tyler to graduate, and Tyler had an idea that he should not cut his hair until his PhD was awarded. And so thank goodness this happened a couple of years later from the standpoint of getting his degree. But there he is, and we know that Tyler has gone on to succeed at what I call the 11th UC campus. DUC, Dominican University of California, is almost a kind of a sister-brother place. And that's where Tyler is up in San Rafael. And you might think that Sarah was kind of related to Tyler. Their hair was kind of reddish. But that's where it stopped. They would sit at opposite ends of the room during group meetings for discussions. But at any rate, these folks were PhD students at that point. Katie Watts is here, and I'm surprised that Katie is here. Laura's here. Laura should have been in our group, but by the time Laura came to campus, I'd stopped taking PhD students, and she wisely decided to come at any rate and work with Roger Lennington. There were postdocs in the group as well. Laurent Calcul. Laurent is now at the University of South Florida working with Bill Baker as a project scientist. There is, let's see, Kim White. Kim is now an associate professor at Humboldt State University, and next to her is Taro Amagata. Taro was a postdoc in the group at that point, and he is at San Francisco State. And Brent Rubio, Brent was a graduate student, and Brent has gone on to be a faculty member at the University of Hawaii system. He came from the islands and had to go back. And then there's Claudia Boot, and Claudia is now at Colorado State University in Fort Collins. So you can see over the years, the group was comprised of energetic, smiling, happy people. And the problem further is that when you run a research lab, is that people come and go. So here's a recent picture. This happened in 2017 of Halloween. All right, so I always like to wear shorts and Hawaiian shirts, and I didn't wear shorts today. I found a pair of long pants in my closet, so that part's working pretty well. But this is the way I come to campus every day. And so the group decided we would have a Phil lookalike contest at Halloween. And so Mitch went into my closet, and where's Mitch? There's Mitch. Wow, how could we miss Mitch? Mitch is our middle son, but Mitch has run our microbiology lab for the last 10 years. He found all these shirts. I'm not sure where everybody got shades from. And then we're sort of toasting our victory. We were toasting prematurely because we did not win. Somebody else won. And so at any rate, it's again, hard to get a picture of everybody together in one group unless you're really expert at Photoshop. So more recently, what we did is we had a going away party for Sonia. And some of these same people are here, but there's other folks in the picture as well. In fact, in principle, all right, so this tall fellow in the back is Jack Lee, and these are Jack's kids. And I'm eyeballing them, thinking that when they get to undergraduate school, we'll be able to get them to be co-workers in our lab as well. So now, let's begin to talk about some nuts and bolts. All right, what are natural products? How can I define what a natural product might be? So when I'm looking for a definition, I always go to the textbook that my colleague, John McMurray, who was here as a faculty member when I arrived at UCSC, he eventually went on to a great career at Cornell University and a great writing career as well. So in the glossary of John's book, he says the natural products are a catch-all term generally taken to mean a secondary metabolite found in bacteria, plants, or other living organisms. So let's think about for a minute some factoids. I'm just trying to kind of lay the groundwork. So from the standpoint of introducing some more molecules, we have a sense of what natural products are. Now, they're not primary metabolites. They're not the things proteins, of phospholipids, of polymeric carbohydrates, or of DNA, RNA. So that's what they're not. Natural products are usually species specific. And I'll give you an example of that in just a moment. And a tool that we use, because our library really is great from the standpoint of subscribing to important literature searching things. This thing is called the DNP, the Dictionary of Natural Products. And if you search the Dictionary of Natural Product in terms of how many entries are there in the natural product area, there's over 300,000, and it continues to grow. There are other search tools that you can use as well, but this is one of our favorite. Now, here's a question. What popular natural products, so NPs, again, natural products, are very attractive to folks living in 10 states plus the District of Columbia? Let me give you a hint. Does this help to clarify things? Maybe so. All right, so you don't have to look at the molecules. If you just see this signal, this sign, you're going to know you're going to go to a place where there's going to be some incredibly powerful natural products. And so this is the molecular structure of CBD. And so, in fact, on the Today Show this morning, they were talking about how CVS markets and Whole Foods soon will probably have deals on CBD-infused goodies. And that will work because the cannabidiol has this molecular structure, which is different than that of the trans-delta-9 tetrahydrocannabinol. So if you make a few small changes in the overall molecular structure, the biological activity continues to change. And that's an axiom. That's a thing in terms of doing natural products chemistry that we're all trying to search for. It's trying to make a connection between a structure that looks exciting. All these structures look exciting, but from the standpoint of the physiological effect, this is the most exciting. In other words, the delta-9 means a double bond right there versus a delta-8 double bond here. That's the cannabinoid receptor firing molecule. This guy doesn't work. And this can be essentially put in candy that you give to your friends and relatives. So at any rate, it's a powerful thing from the standpoint of how a small change in a molecular structure will cause the overall suite of biological activity and physiological properties to change. All right, so let me just dwell a little bit further. And again, I apologize that there's so many molecular structures here. But again, for those of you that are not so much interested in the interconnectivity of all the different carbons, just look at the molecular shapes. And just think of this as an exercise in pattern recognition. All right, so natural products have been incredibly valuable for society since antiquity. In fact, salicylic acid. There were no takers for the aspirin tablets, or I guess I never gave the ushers my bottle. I forgot to do that. But at any rate, salicylic acid has been used since antiquity, and of course it comes from the bark of the willow tree. And so for years, this was the form until the Bayer company stepped in and added some more appendages to make it a stable, formulatable pill. Now, the point here, in terms of all these molecules, all these things that are useful in our pharmacopoeia come from natural sources. Most of these things are plants. Morphine is the example that propelled Merck into profitability in the 1800s, and you can see its molecular structure here. The problem is with these opioids, we've got a challenge from the standpoint of the next generation of pain medicine. And so this complicated ribbon structure, it's a polypeptide. This is a compound called Pralit that comes from, I've got the name wrong, it's zirconolide. This comes from a cone shell, toxic cone shell. So this is an alternative from the standpoint of pain medication that doesn't have the addiction properties of morphine. Penicillin G is well known to all of us, and my point here is that penicillin G helped the U.S. to win World War II. Taxol is a compound that was introduced in the 90s to cure lots of solid tumors, and we can consider this to be a wonder drug that came along, as I say, many decades after that of penicillin. In 2015, Nobel Prizes were awarded, again, for work that people had done on two important natural products, not shown here, artemisinin, an anti-malarial, and the other compound was ivermectin, a compound that is an antiparasitic. So, as I said a minute ago, there are over 300,000 known compounds that you can get when you go and search the Dictionary of Natural Products. And what I haven't begun to tell you about is this less explored frontier of the marine realm. So what I want you to do is to stay tuned. There's some more stories coming. I'm not sure if Scott Loki couldn't be here today, and I'm not sure if Cam and Josh were here from their new company that was just established on the west side. But these folks like to wear a hat that says the word Bro 5. And Bro 5 is a rule I'm not going to talk about, which most natural products fit. It's called Beyond the Rule of 5. And this is a construct that most medicinal chemists would say has to be present in a molecule for it to have reasonable biological activity. In fact, I'm sure the Rule of 5 is still practiced in terms of the med chem that goes on at Roche, not Roche anymore, it's Genentech, and down the road Gilead and Exelixis, et cetera. But at any rate, so there's an emerging sense on our campus of people walking around with Bro 5 hats. So if you see anybody with this hat on, you'll know what they're talking about. Now, so let's go back a little bit, because, again, I've totally strayed from the mission that Professor Whipke articulated, because I never really looked at the email to see that that was something I needed to do. So at any rate, here's the circumstance. In the early days of natural products, there were these meetings that happened. Drugs from the sea. So in '67 at University of Rhode Island, people got together to talk about this new emerging initiative and opportunity. We're going to have all sorts of incredible biomolecules from the ocean. And so the meeting happened. The cover of the book was pretty interesting. I was able to get pictures of these covers because my friend Francis J. Schmitz at University of Oklahoma maintained a fairly good library, although the book from '76 was a little bit mistreated. You can see that there's some things missing from the cover. But 9 years later, there's some problems. The title's been changed from Drugs from the Sea. It's Drugs and Food from the Sea, Myth or Reality. You know that food comes from the sea. So they were kind of fudging a little bit. And now what they've got is, instead of beakers and things, we've got marine critters, we've got seaweeds, we've got a molecule that has bromine, chlorine, and another bromine. And so it took 9 years just to make that one little leap. So you can see in the early days, the field moved pretty slowly. Well, so in those early days, I was here, and I was beginning to pursue, it seemed like the thing to pursue is things that were like on that cover. If you can find molecules that have chlorines and bromines, this might be the ticket to fame, to fortune, but also to tenure. And so, in fact, again, I think the thing, it's not hard to spot the Cl, that's chlorine, the Br over here, that's bromine. And so there are a number of papers that we had. And here, this is late 70s, early 80s. We're sitting in the laboratory. This is a typical laboratory in that period of time at UCSC. No fume hoods. Some people don't even need to have lab glasses because there's no sense of needing to worry about an EH&S, an environmental health and safety officer, because they weren't appointed yet. That concept hadn't come. The other thing that was important was not to have your hair so long that if there were a Bunsen burner going, there would be a little fire. And so, anyway, that's Larry Maines. And Larry, not only was he a long-haired dude, but he had a giant stash. And so Larry had all of the kind of trappings of what it meant to be a scholarly graduate student. I tried my best to grow long hair. It didn't work very well. And I could grow long sideburns. That part worked. Plus, if your hair got too long, you're not going to have the respect from your other colleagues in terms of thinking that you're a fine scholar. So, anyway, the journey continued. We wanted to really look at sponges. That was the initial area of inquiry, and I'll get to that point in just a minute. And in fact, we thought we were on the right track when we had an expedition to the Caribbean, and we collected something that looked like a sponge. It turned out to be a soft coral, Briareum asbestinum. And so it was a hard lesson to learn that all this chemistry we were after was not from a sponge, it was from something that wasn't a sponge. Now, one of the things we tried to do is to go beyond our front door for expeditions. So this is a picture from '77, and here I am. My hair's getting maybe a little longer, but nothing in comparison to these individuals here. This is David Sherman. David was an undergraduate in our lab, and David is now a full professor.
Which one is David Sherman?
This is David Sherman. You can't tell? And then that's Rick Hogue. So, yeah, David had this frizzy curly hair.
Can I take a picture of that?
And Tyler, I'll send you the picture. All right. And this is Rick Hogue. So we're on our way to Oregon to collect more Procamium oregonium. And we stopped for a bite to eat. If you could see the sign here in terms of, it was cheap to do expeditions in those days because you could have a hamburger stop, hamburgers were 50 cents. And I wouldn't spring for an extra quarter so we could get cheeseburgers. Guys, we're going to have hamburgers, and that's it. And we're going to keep moving forward. So that's all well and good. But the real paradigm of where we were trying to head in our research was to look at marine sponges. Here is an axiom in natural products chemistry. The simpler the organism, the more complex the chemistry is. And so that's why we have, this is my view of why we have so many important biomolecules from microorganisms, from gram-positive microorganisms, fungi, et cetera. And you've seen some examples of these things. And I'd say the molecule diversity outstrips that of the phytochemical world. And I'm glad there's no phytochemist here because there could be a big battle outside from the standpoint of the discussion of this. But anyway, all right, so my idea, let's take this view of the different phyla from the marine realm, and let's focus on the simplest organisms. So we began to look at red algae, the Rhodophyta, but really what I wanted to look at were the sponges. And so that's the simplest of invertebrates. And so if this paradigm were true, the simpler the organism, the more diverse the chemistry, searching and exploring sponges could be a gold mine. All right, so let's just talk a little bit about sponges. They're primitive. They're the most ancient of organisms on the world, known to the world. And remember that point, because it's going to become important in just a minute. They have an aquiferous system. They pump a liter of seawater per cubic centimeter of tissue per hour. So they have this, what's called an aquiferous system. There's no tissues, there's no organs. The form is very plastic. So in other words, that says that the sponge you collect in area A might be the same as what you collect in area B, but the whole morphology could be different. And so that's going to confound a little bit the business of trying to recognize and interrelate species that could be related or could be not related. The analogy to stem cells are what are called totipotent cells in sponges, and incredible biodiversity. All right, so now here's a quiz. We've decided that we want to go and we want to look at marine sponges. So let's interrogate each of these panels to figure out which are the real marine sponges. What are the things worthy of our attention? Somebody's calling about SpongeBob. Yeah. No, I can't talk right now. I'm busy. So, SpongeBob. Why isn't SpongeBob a real marine sponge? Well, firstly, it's got a funny morphology. It was designed to mimic a kitchen sponge. So it looks like a kitchen sponge. And also, it really came on the scene in '99. That's when Nickelodeon started having the SpongeBob things. So this is not a sponge that's ancient. It's a more modern sponge. So there's two strikes against SpongeBob in terms of being a marine sponge. Professor Finnicle could probably tell us what species of Gorgonian this is, but maybe not. I'm sure Bill knows all about these things. I won't attempt to assign an incorrect name because I'll get into trouble a little bit later on. But it's clear it's not a sponge. What about this octocoral, this sea pen? No, that's not a sponge either. So we only have one thing left. This must be a sponge because it has one of our collection tags embedded in it, and it's got the photograph. So there's a sponge. That's great. All right, so we have a good sense of how to find sponges. You go diving, and you look for things that have collection tags embedded in them, and those are sponges. All right, now, along the way, Christina Diaz was here as a PhD student, and Christina and I have maintained contact for many, many years. And so let's look at what a sponge is through the eyes of Christina. And so notice, University of California, Santa Cruz, if it had a sense of what, her degree was in oceanography or ocean sciences, but really I helped Christina to land here is because the interaction, the love of sponges that Christina had, and my love of loving the sponges that Christina could show me, and then exploring the chemistry. So Christina's going to help us now to really get to grips with what a sponge is and how to recognize it.
To make the invisible visible, Diaz injects a harmless colored dye into the water near the body of a sponge.
One of the ways we can test for the rate of water flow that moves through the sponge body is to inject a colored dye and measure the speed at which it is pumped through the sponge. When I start seeing the colored dye coming out of the sponge in less than 2 seconds from when it was applied, I could not believe my eyes. This steady and strong continuous flow of water continuously coming out of the sponge was an incredible realization of the dynamic existence of this organism.
The Unella swinohi, the sponge we saw a few minutes ago. This will be important later.
It's an incredible sight to witness.
So if you want to find sponges, you just need to do two things. You need to take Cristina Diaz on all expeditions with you, or take a syringe full of fluorescein dye and squirt it into everything you might think is a sponge. Well, eventually we learned how to recognize these things from the standpoint of looking at the natural history. And so we stopped taking Christine with us, and we never took fluorescein dye with us as well. And we're pretty successful from the standpoint of developing a fairly large collection of sponges. And it's important to describe and sketch what this is, but I've got to wait a few more slides to do this. Now, something that happened in the early 80s, from 1980 to 92, there was something called a California Sea Grant Program, Marine Pharmacology and Chemistry. And what it did is it brought 4 investigators on 3 UC campuses together to engage in discovery and pharmacology, all while talking and interacting with each other in presumably a collegial way. Now, so one of the projects was here at UCSC, molecular tools from sponges. The other, the pharmacology group, was on the UC Santa Barbara campus, and the late Professor Bob Jacobs led that. There were 2 projects on the UC San Diego campus. The late Professor John Faulkner was looking at sponges and anti-inflammatory leads, and Professor Finnickel, who I mentioned is here, his early work dealt with looking at anti-inflammatory compounds from soft corals. And in fact, the pseudoterracins were the things that Bill and his coworkers discovered. And today they live on as an active constituent in some of the Estee Lauder face creams. Now, to get a sense of how important my view was of the various teams, I've numbered them from 1 to 4. Notice, 1 is a little bit bigger than the others. And so it was important as to what the other people were doing, but it was important as to what was going on the UC Santa Cruz campus. And of course, we were further away. We were in the north, buried in the middle of the redwoods, and so we had to do something to stand a little taller. So the only thing I could do was to make a big number 1. All right, so as was mentioned, Katie, in the later years, during her PhD, looked at jasperkenolide, but the initial discovery of jasperkenolide came from Larry Maines, and we published a paper in 1986. And in fact, at that same time, the Faulkner Ireland team found the same compound and also published a paper in that same year. They gave it a different name. So sometimes these things happen from the standpoint of just the names out there will be the same molecular structure, but a synonym. And so at any rate, I've continued to enjoy using just the name jasperkenolide to describe that compound. And when I go and do a literature search, that's what I look for, is I look for papers that are citing jasperkenolide. And so if you look from our early publication to, as time went on, more and more papers came with this molecular structure as a kind of a seed for the science. And the creamy bars are the chemistry. In 95, with the people of the NCI, we found that there was therapeutic potential and anti-actin activity for jasperkenolide. And so that sparked a whole bunch of biological studies. And over the years, we've continued to supply other laboratories and the like with compound so that they can essentially have these in their armamentarium. My colleague Joe Konopelski is also responsible for one of these bars. I can't remember where it is. And Joe was doing total synthesis of jasperkenolide as well. And now, so the other day, let's see, today is the 9th. So 3 days ago, I began to think about, I better try and do something to answer Todd's thought about just the impact of science from the later years. So I did a literature search on not SciFinder, it was the Web of Science. And I simply asked the Web of Science, tell me how many papers have been published with the word jasperkenolide as a topic. And so to date, there could be a few more now, because I didn't check it this morning. There's almost 640 papers that have happened. But what's also interesting to note, I'm not sure if you guys talked about the year I became an Emeriti thing. But since 2012, there are a bunch of papers that have been published. We have a few, but other people have also published papers. So in 2019, just as of this month, there's 6 papers. In 18, there were 25 papers. So after 30 some odd years, this compound is still an important thing that motivates labs, including ours and elsewhere, to explore that science. Now, as our program developed, so that marine chemistry and pharmacology thing was important. And when that program ended, it was a bad day, not for the guys in Southern California, because they were good at getting money, but it was a bad day for me because I had to figure out how to get money in other ways. And so part of the strategy, part of the challenge was let's get our group really moving forward. And to do this, there were several steps. So we needed to expand the marine-derived taxa to sponges beyond some of the areas in the kind of eastern South Pacific. And more of that to come in just a few minutes. But what that involved was organizing expeditions, finding scientists in host countries that we want to go to, learning how to operate in areas that were pretty much the outback, but also managing the UCSC team. And sometimes folks would want to play tricks on me and give me drinks that came from boxes that were labeled as vino. And so that was another sort of challenge. What we had to also do was to connect the natural history of these sponges, because Cristina Diaz could not be with us on every expedition. And so we needed to learn how to make this sort of feedback loop. We needed to learn how to be more efficient in organic structure analysis when we brought samples back to the lab. And we needed to then, as I said a minute ago, set up the funding and the network for looking beyond the straightforward organic chemistry. We needed to connect up with people involved in experimental therapeutics, because if we were going to make the connection between a therapeutic lead or molecular probe, we needed to have that sort of interaction. So over the years, it took me a long time to learn this, but if you go talk to marine biologists and ask them where is the center of biodiversity, they'll tell you it's this magic triangle from Singapore, from the Philippines to Singapore to New Guinea. And this, eventually, what I decided, this is where we needed to be doing expeditions, in Indonesia and New Guinea. And so eventually, that became a possibility. Along the way, we set up a repository for our sponges and for microorganisms, both fungi and gram-negative microorganisms. And so from the mid-'80s to 2018, I reckon we've got over 3,500 sponges. Some have firm taxonomy, some have provisional identifications, but they're all sitting in these bottles in 2 different cold rooms, one in Tiemann Labs and the other in the Physical Sciences building. I figure we've got 1,000 microorganism strains, and what we've been looking at in later years is the 462 Indonesian sponges that we collected during these years. I'll tell you right at the end how we've been able to resurrect that sort of pattern from the standpoint of having access to natural material. But at any rate, we were also able to go to the National Cancer Institute Developmental Therapeutics Program, because they've got, if you simply write a letter and you ask please and you say thank you at the end, they'll send you samples. It's not quite as simple as that, but part of the please thank you thing does work. we developed strategies that might be called, in the old days, they might be next gen. Now they're pretty standard from the standpoint of dealing with these, with all of these repository materials. And so we've got over what I call 8,000 distinct testing units sitting in 96-well plates available for high throughput screening. And we've got over 1,100 pure compounds that have registry numbers in our repository. That's a little bit of an overestimation, because you go look at the tube or the vial that has a compound, and you can't even see anything. And then you take the vial and you try and measure an NMR, you still can't see anything. So some of these things are either empty or they've gone to nothingness. Now, an example of how we were able to connect up with therapeutics is this. My colleague, Fred Valeriot, who's at the Henry Ford Cancer Center, has been busy using a very simple discovery paradigm. Fred takes cancer cells, he takes leukemia cells, takes solid tumor cells, up here, leukemia cells, normal cells, and he puts these in soft agar, and he's doing something that's like an old-fashioned antimicrobial test. Well, he'll take the disc to which compound or distinct testing unit or crude extract has been put, and he'll look to see what the zone kill is from the standpoint of what's happened. And if there is selectivity, in other words, if the solid tumors are knocked off faster than the normal or the leukemia cells, he's envisioning that there's some sort of therapeutic index. Nothing is known from this screen about what the molecular probe, what the molecular action is, what the targets are, but this is certainly a good start. And what Fred has got is he's focusing on ovarian cancer, a brain tumor, PANC1, a liver cancer, and a glioblastoma line that comes from patients at the Henry Ford. So he's got these five cell types that he's using to kind of drive the work forward. Now, an early compound we found that was cytotoxic, and we discovered this from other inferences, is a compound called latronculin A. And again, I just want you to look at all the different parts. There's oxygens, there's nitrogens, there's sulfurs, there's a potpourri of different sorts of things that are present in terms of that molecular structure. So I only have one of these kinds of pictures, and everybody has these in their collection. We saw these earlier from Sarah and from Tyler. I wish we had gotten Steve Claybush to take a bunch of these, but we only took one picture, and everybody has it. So here, what I'm doing is I'm searching the reef. I'm swimming as hard as I can, hoping there's no current, hoping also there's no things in the background, those things in the suits, the dark gray suits, the sharks, right? And so we're collecting, we're sampling, and I'm thinking. So let's explore a little bit of this sampling and thinking. So one of the things that we found that was kind of interesting is this nudibranch. This is Chromodoros quadricolor. And Chromodoros quadricolor is loaded with latronculin A type molecules because it keeps the fish away from this thing. It's not a fast swimmer. And so it protects the nudibranch from destruction by omnivorous fish. But this nudibranch, it's sitting on a red carpet. What could this red carpet be? Don't everybody say the word at once. A sponge. Yeah, of course. Yeah, it's sitting on Latruncula magnifica. And the nudibranch is about as big as your thumbnail. And the sponge is way bigger than that. And so the nudibranch is what I call a spongivorous nudibranch. It's busy having lunch by feeding on Latruncula magnifica, and it's just sucking up Latronculin A, and it's protecting its life, better living through chemistry, better living through Latronculin A. So somehow the nudibranch learned that lesson. Here's another nudibranch. It's even got a happier life, even though it doesn't get the red carpet treatment. Look at the smile on its face. And this nudibranch is Chromodoros loci. And it's sitting on a black carpet. And so what do you think that black carpet is? That's a sponge, of course. Yeah. That's Cacospongia mycophagiensis. And Tyler and many other people in the audience that have been on expeditions with us know this sponge quite well. And they also know how the nudibranch just can't get enough of just sitting on the surface. It needs to crawl in the osco holes looking for more latrunculin A. I'm not sure if the holes were richer in latrunculin A or not, but that seemed to be a pattern. Now, I want to switch gears just for a minute and talk about the project that Madeline started in the 80s. And Madeline, that's that first paper with you and Emilio. And while this research was going on, I can tell the story since Madeline was here, there was a lot of discussion. Sometimes the words would get very loud. There was shouting and screaming. There sometimes were four-letter Spanish words, because Emilio was from Spain, and Madeline was from, no, she was from the East Coast. Yeah, she swore in a kind of a Northampton type of New York accent. And, but at any rate, so this started the train. We discovered the binghamide series, and there's been many, many subsequent papers on the bingamides. And the thing to impress upon everybody is once we had the discovery in hand, we had to go back and collect more sponge. So we go on an expedition, and I tell people, go look for an orange sponge. And so most people will come back with the thing, that Cal, you remember this thing, the WPO. It was everywhere. It had no bingamides. It had nothing of value. We call it the worthless Pacific orange. And so at any rate, learning how to collect this sponge was a key. And if you turn the thing over, you always found these white crustaceans. Sometimes over in New Guinea or in Indonesia, it wasn't quite that way. But in the Fiji, Vanuatu area, that was the case. Well, this compound continued to be important in our lab because in our collaboration in the 90s and 2000 with Novartis Institute of Biomedical Research, they found that the initial solid tumor activity we discovered was really very robust and very specific. And in fact, some analogs had oral bioavailability. So we engaged in a campaign to recollect the sponge, to look at analogs. We must have looked at about 50 or so analogs. Zia Thales' PhD dissertation was on that. And the people at Novartis were busy synthesizing analogs. What they found is that the warhead, or the pharmacophore, or the thing that gave the anti-tumor activity was the center part. You couldn't mess with any of those pieces. The caprolactam had to be there. It couldn't be a 6-membered ring. It had to be a 7. You could mess with the ester group, and you could mess a little bit with this part. But you had to have this double bond in this sort of trans relationship. In other words, this part down here and that part up there. And if you switch that, then the activity went away. So this compound entered a phase 1 clinical trial spurned by the Novartis people for a couple of years. And it was against phase 1 patients. And unfortunately, it came to a close after a couple of years because there were some cardiotox problems, and there were also some questions about the molecular target. And so to move things forward, the toxicity issue and the target issue had to be examined. But when Madeline was busy with Emilio looking at the structure, it took 3 or 4 years to get the entire structure. And this was before, well, what I show you here is Tyler, he bought the old, for his slide, the first edition, which you can get pretty cheaply now from Amazon. I guess the second edition is not much more expensive. But at any rate, this was presumably the Bible that everybody that came through the group would use from the standpoint of doing structural characterizations. Well, in fact, Bill Finnicle, I think you're responsible for this. You talked at a meeting about the Cruz rule. You talked at a Gordon conference. And when you say something at a Gordon conference, nobody's supposed to repeat that. I didn't invent this, but Bill did. And then other people jumped on this. And I didn't define it. I did define it because of an accident we had. But today, if you go and search on Google, Cruz rule, you come up with a bunch of articles. And it tells you when the ratio of protons to carbon and the sum of other heavy atoms falls below 2, an axiom that has been dubbed the Cruz rule comes into play. Well, I began to see this in the literature. I never articulated this rule as, I said what it was, but I didn't give it a name. But it was great to see the name, except a little bit later on, here's another paper. Using HMBC and adequate, well, let me just get to the point. Is the Cruz rule obsolete? So this hurt. I had to go home and tell Peg and Mitch and Neil and Darren, you know, our name has been dropped up and down the sidewalk of chemistry. And so a couple of years ago, we were able to come back in a paper that was published in 2017 to attack the people that talked about this. This is the people at Merck that were besmirching our family name. Shame on them. Well, there was reason for this. All right, so PR. Is there a person here with those initials?
Please stand up.
Where is that person? There he is. He's sitting next to Karen. Paul Relifo. Paul did his PhD, and he had some tough assignments. He was involved in looking at the structure of spiralosetidine. And fortunately, Paul finished his PhD before the nasty emails started to come. And it wasn't Paul's fault. It was our fault. You know, we're partners in crime. Paul was gone. He fled the scene, so I was getting these emails. And they said that there was a problem with the structure. And the ratio of protons to carbon is 0.6. Now, ergogoline is a plant-derived compound. Its structure, the center core, has that same problem, but X-ray crystallography was done to solve that structure. Paul couldn't grow a crystal because, by and large, crystals of important molecules don't grow in the middle of a redwood forest. Had Paul taken some of this with him to BC, isn't that right? Or is it BU? BU. All right, if you'd taken it to Boston University, a crystal would have grown. And eventually that happened. But at any rate, Paul, you need to get this thing because we've got a 0.057. Nobody has questioned. There have been no nasty emails about the structure of leucoselenamine. They may be coming next week. and so, but at any rate, all these compounds violate this rule. And so what I say is you can't do it by NMR. You need other things to solve a structure. So Danishevsky, Sloan Kettering, Cioflino in Canada at UBC, and Watson in Australia, all were sending me emails saying, there's a problem. Our beautiful structure is tarnished. That's, if you read between the lines of the nasty things they said in their papers, that's what you come up with. All right, so the problem, all right, spiralosetidine, the structure developed a smell, a stench. So what was going on? So this is Danishevsky. They were hurling stink bombs to the UCSC gang. All right, so in the middle, originally I was in the foxhole. Paul had left, and so Taro Amagata and Kem White had joined the group as postdocs. I showed you their pictures earlier. And I convinced them to take my place in the foxhole. So you can see there's the UCSC banana slug flag, and there's our problem with the H over C less than 1. But nonetheless, the stink bombs are coming from New York, from Canada, from Australia. How are we going to solve this problem? All right. So what we needed to do was we needed to do more reisolation and data collection. And I'm not sure if I ever invited Paul to come back for that part. No, he was busy making money and becoming famous at Boston University. And so we knew eventually that there were some things. The things in red were problematic. And again, it wasn't Paul's fault. It was Paul's fault and my fault. And so we're hoping that Kim and Taro can come to the rescue. All right, so again, over the years, what the job is to motivate the group to solve some tough problems. So this is a problem, and it gives a structure elucidation challenge. So I came up with the acronym CAN. All right, so CAN means you got to choose the correct structure. That's the C part. You have to assign chirality, and you have to do that in a proper way. And you also have to get new samples from the repository. All right, so that was potentially possible. And what we needed to do was to essentially come up with the bit of no more red in terms of the data set. But it took a while to convince Kim and Taro to participate. You would have thought that I was telling them that this is what you've got to do. And the quote here is, and you thought your job sucked. Right, no, I'm not asking them to collect elephant poop. We'll just get some samples from the repository, and let's move forward. So the structure was revised. We were able to do fancy calculations. We were able to get that X-ray crystallography sample and the data collected, and we did fancy NMR thing. And so we eventually were able to solve the problem. And you can see the red stuff is very different than what the eventual structure turned out to be. Again, just looking at the patterns of the oxygens and the nitrogens, you can see there's differences. Here is a nitrogen, and the correct structure had an oxygen. Small but significant differences from the standpoint of things. So once that happened, the hate mail stopped. Once we got the structure right, then we were able to move forward, and we were able to move on. Tyler has stolen my thunder, or was it Sarah? This movie Medicine Man is just a great thing. Once a year, everybody should go watch Medicine Man. And what you see when you see the movie is that it involves mass spectrometry. So this, Sean Connery plays a guy, Robert Campbell, who is a biotech person from the East Coast that's in the middle of the Amazon jungle. And so what goes on, this is kind of hard to read. Let's see if I can read it. Quote, in terms of the leader for the movie, he turned his back on civilization only to discover he had the power to save us. But I interject by using mass spectrometry. And that's what was going on in this movie. He had a mass spec out in the middle of the Amazon jungle, and he was making incredible collections in that regard. So this is one of two of my favorite movies. The other favorite movie is a movie called Bottle Shock. And that involves kind of wines. But my favorite paper when I'm watching Bottle Shock is this paper that also involves mass spectrometry. And Sarah, you guys need to take back to your group meeting a sense of the question, do white grapes really exist? It was published in the Food Research International Journal in 2015. And I'll just give you a hint. Mass spectrometry is used to answer this question. So if any of you want this paper, I can send you the PDF, and you can explore it on your own. And it's an exciting circumstance in terms of mass spectrometry helping to answer this almost philosophical question. Now, what I believed many years ago is that new frontiers would happen through using mass spectrometry screening by a technique called direct analysis in real time. It's called DART. And here is an incredible proof of principle result. The way the machine is set up, it's like doing atmospheric ionization, where you can put in the source where the ions are being created and then analyzed in the detector. You can put things in the source to analyze. You can put chemical samples. You can put dollar bills or $10 bills. I first learned of this when I went to the University of Pacific a number of years ago to give a talk. And I went to the mass spec lab that was run by this fellow, Professor Sparkman. And he said, I've got one of these DART sources. And I know you come from Santa Cruz, which is laden with crime and mayhem and madness. Let's put a Santa Cruz $10 bill in the source and see how much cocaine is on the surface of that bill. And let's also look at a bill from Stockton. And what we're going to look for is just the ion that's created from the standpoint of having that $10 bill in the source. And so if you know how to interpret the data, you can say, all right, yes, there's a fair amount of cocaine. Turns out the Stockton dollars were way worse than those of Santa Cruz. And I was vindicated from the standpoint of seeing this. But it was an important thing. So I went back home, and we bought a DART source, and I convinced Katie to look at halogenated metabolites from invertebrate-derived fungi. And Katie did an excellent job, and the paper's published. I tried to convince Tyler to do the same thing. So we're looking at specimens of Cacospongia mycophigiensis, and we found some really interesting results. That was in 2012. Do you notice this point here? The paper was never published.
You also spelled my name wrong, so keep it like that.
Oh, yeah. That's the problem. Okay, maybe that's not you, Tyler. That's somebody else. Yeah. At any rate. But Tyler, please go home and finish this paper. Yeah. Now, there's Tyler. His name is spelled right. His name is spelled right. His name is missing from that, and his name should be there probably. But at any rate, that's a cruel world, Tyler. So here's an interesting thing. Tyler helped to lead the campaign to look. We saw this sponge when you go to Fiji and you find the sponge with the nudibranch on it that has Lotrunculin A. It's called Cacospongia mycophigiensis. As you begin to travel through the 17 south latitude of the Indo-Pacific, you find other versions of the same sponge. Remember earlier I said sponges can be very plastic and changeable in terms of their morphology. The key to us eventually was the same nudibranch that was on the thing in Fiji is on this thing from Vanuatu or Solomon Islands. And when you go to New Guinea or Indonesia, you see that same nudibranch. So there's three different forms, three different morphological forms. And when all the shouting was done in terms of the chemistry discovery, we found that there were 5 different chemical forms of this sponge. And it seemed kind of crazy. We knew the taxonomy. What the nudibranchs were all after was Latrunculin A. They were after, where's Latrunculin A? There he is right there. And it turns out that a turning point in the Latrunculin A research was a paper that we interacted with Dave Rubin, cell biologist at UC Berkeley. And he found that Latrunculin A was also a powerful actin inhibitor. And since the publication of that paper in '97, this paper has been cited almost 600 times. And so it's been an incredible sort of thing in terms of the discovery. By the way, we were not the first ones to discover Latrunculin A, but we discovered a whole bunch of other analogs in all these other compounds as well. And so the question is, what's going on? Are there microorganism communities that are responsible for such a large variation in chemistry from an individual sponge whose taxonomy we know? It's all been verified by Christina Diaz. Well, all right, so sponges are communities. There's a microbiome. There are gram-negative bacteria. There's gram-positive bacteria. There's special bacteria that are only found on sponges called Porreobacteria. And this work comes from papers published by 2 outstanding scientists, Ute Henschel and Suzanne Schmidt. And what they did is they were using culture-independent methods. They're using essentially molecular genetics, focusing on the 16S ribosomal RNA for sequencing. And so they're able to then pinpoint that in a sponge, boy, there's a lot of different things that are present from the standpoint of the DNA. But this doesn't tell you anything about what you can culture from the sponges. It just tells you what's there. So that's been our goal. And I'm coming back now to the bingamide bit. Here's the structure of the bingamides, as Madeline described it way back and as we described it in other papers. In 2000, what was it? It was about 2010 or 12, something along these lines. There was a patent from Sanofi Aventis. And what they said is that they found an organism from the dirt, and they didn't quite tell you where it was, that could produce not only this guy, come on back, whoa. Where is it? No, there he is. It could produce the bingamide E, but also something called bingamide E prime. So eventually we were able to get that organism and grow it in our lab. And the organism turned out to be Myxococcus virescens. And so it reliably gave us bingamide E derivatives. In fact, what you can see is, this is data dense. There's a number of people, past co-workers from the lab, and if I didn't show them this, they would wonder if we're not being correct. But if you do extracted ion chromatography on the culture plate from Myxococcus virescens, you can see both bingamide E and bingamide E prime. And if you look at the molecular weights, the 358 and the 372 are augmented by H+, because this is electrospray ionization mass spectrometry. Sorry for these complicated words, but that's the data that was so needed to verify that we were able to do this. And so the circumstance was this provided a way where we had this biologically active important compound. If we wanted more, potentially we'd just go back to our lab. We didn't have to go back to the environment to gather more material. So with that as the kind of jumping off point, some years ago, about 6 years ago, this was during the Emeriti period, we got an NSF grant, and we were able then to look at this workflow from the standpoint of taking both soil and sponges and eventually getting gram-negative bacteria, doing a bunch of fancy things, and eventually getting molecules. That was the goal. So what we did is we divided the state into 5 different areas, and we proceeded then to do expeditions. In fact, young Mitch here was the one that led many of the expeditions to the northernmost point, and Mitch and Matt went down to Southern California to also get soil samples. Basically what we're doing, we're getting sediment samples from the ocean sand interface, from the drier sand, and from the sand-land interface. And so we had all these things, and we put it through that workflow, and we're able to get lots of different gram-negative bacteria. We're after really myxobacteria, but we weren't very successful in that regard. But one of the things that was intriguing that came out of this is when we used that workflow to examine one culture specimen in particular. This was an organism taxonomically identified as Pseudomonas benzoniferans. And what we did is that if it was grown in a specific media, we then were able to see clusters, again, through a networking pattern that was developed by, in fact, Laura Sanchez is the resident expert at doing these networking patterns. And so whenever we would have a question, we would call Laura. And Laura's very generous at helping the gang in the lab to figure this out. What was exciting is the network, in terms of the mass spec, told us we had compounds called Thionella peptides. Thionella was a sponge that I talked about earlier. And the molecular structures of these things are here. So when we grew the first grow out, we saw from mass spectrometry the base peak of these compounds. We did another grow out, and we saw again through extracted ion chromatography base peaks. We never had enough material to do an NMR. We published this in connection with a bunch of other things, but we told the world that basically for a fleeting moment, we had something that looked like a sponge metabolite. But we warned people we cannot send you Pseudomonas benzoniferans and have you repeat our work. This is a continuing theme from the standpoint of people isolating compounds from both gram-negative and gram-positive sponges and growing them in culture and then having that culture kind of go haywire. So kind of to begin to wrap things up, the abiding interest and the continuing interest is to look at the biosynthetic origin of sponge-derived products. And I've told you at length about the bingamids. We've talked a little bit about jasperkinolide. And if you look at jasperkinolide, there's a sponge. There's the compound jasperkinolide. This is a compound that comes from a gram-negative bacteria. These compounds are called murianimids. And they're very similar. And what we believe is that we someday will be able to isolate the gram-negative bacteria that produces the bingamids from the bingamid sponge. We haven't been able to do that yet. Someday we'll be able to isolate the gram-negative bacteria that comes from this particular sponge as well. And we've got several other sponges that are in the background. So in the expedition in 2017, that is part of our collaboration with the Diponegoro University called UNDIP. They're in Samarang in Indonesia. We were able to collect a bunch of sponges that were targeted, and you can see the kind of inventory. We got samples, and we brought them back to the lab, and we've begun to try and work out methods to isolate the gram-negative bacteria. To expand the frontier, the hour's getting late. Tyler, you guys took too long for the introduction.
It was 7 to 8 minutes.
Any rate, Sembrin is a compound that we've been engaged in pursuing the chemistry of for years, and we wanted to get more of either the producing sponge or the microorganism producing sponge. Paterin is a compound that comes from a terrestrial beetle that's hypothesized to be produced by a gram-negative bacteria. And so the reason Sembrin is so important is it's got nanomolar activity, and it's got a position where we can make an antibody-drug conjugate to capitalize on that biological activity. All right, so in the 2018 expedition back to Indonesia, I primed the group. All right, if you go and collect the sponge that produces Sembrin, in fact, you know all these are sponges, especially these upper ones, because they have the little collection tag thing in the picture. But the only one that's worth anything is this guy here. So what the group had to do was to go and not worry about this guy, or this guy. We wanted this one. And, well, the whole thing kind of, it was exciting for a while. We went back to Halmahera. This is the Sulawesi in Indonesia. Manado is up at the northern part of the island. If you keep going north, you get to the Philippines. And so the group got on a boat in Manado, journeyed across, spent time in Halmahera, spent a lot of time at Ternate. And the group was enthusiastic. There's Mitch, there's Aaron, there's Cal, there's Mary Lou, and there's Rick. And so they were all busy trying to find sponges, but unfortunately, we came back empty-handed. We came back with other things, but none of these. Again, it shows you, remember back in 67, the drugs from the sea thing, how long it took to just put a brominated and chlorinated molecule on the cover of the proceedings. Well, this is a long journey. So in closing, from the standpoint of just giving you a sense of where things are going, this was a review that was published in 2012, and it talks about natural products as being a major source of drugs. And so if you look at the timeline, and way back here, Sarah told me earlier, she could read that number, which is 50.0% of the pharmaceuticals were based on natural products or analogs or whatever. And that pattern has continued over the years. This charts the progress from 1981 to 2010. So I'll leave you with the prediction is that the future prospects of sponges and gram-negative bacteria are going to be the therapeutics that are going to come from 2020 onward. Well, let's hope for the best. A final conclusion, our Charles Schultz people tell us, please remember natural products are awesome and vital to advance the frontiers of science. So let's keep this in our mind's eye. And, well, there's a lot of names here. And Tyler and Sarah got it mostly right from the standpoint of we've published to this point 242 peer-reviewed publications. There's a couple more that are in the works that are kind of weaving their way through review and the like. And along the way, there were many PhD and master's students. And so we already know about Sarah and Paul and Tyler and Claudia and Nate Seagraves. I'm not sure if Nate's here. We know about Christine. I take credit for Christine because I helped her to get here from the standpoint of her PhD. There's a couple of people that have asterisks. Wayne Enman. Wayne was a PhD student here. He was a postdoc. Somewhere his name is there with an asterisk. And he was a staff person. And there's Madeline was also briefly a postdoc and also a PhD student. And we won't go into what caused that sort of return. But oftentimes people return back to the scene of the crime. Taro was a postdoc here twice. He left and he just couldn't stand the pain of not being in Santa Cruz. So he came back as well. So thanks to the people that are really important in terms of being able to do this. My wife Peg, our gang, the diving professionals, Cal and Clay Weissman. In fact, Clay was responsible for this picture. All right, so Todd is about to oust me from the podium here. I'm ready to go. I'm ready to have coffee and muffins and whatever. So we've reached the end of the journey. Thanks very much.
Thank you.